Thank you, I just got a recording pop up. Thank you, everyone, for joining us at our B-CRATOS webinar. I think this is the 5th one of these we've done this year. And today we have Professor Hans Scherberger joining us. He's a participant in the B-CRATOS project. I'm going to give a brief introduction for Hans and then let Hans talk to you about some science. So Hans received his education, master's in mathematics, and he has an MD as well at the University of Freiburg in Germany. He's currently heading the neurobiology lab at the German Primate Center and is also a professor for primate neurobiology at Göttingen University. And he's been there since 2008. He has trainings in systems electrophysiology from the University of Zurich and also at Caltech, where he worked with Richard Anderson. He also led a research group at the Institute of Neuroinformatics at Zurich University and ETH. His research focuses on neural coding and decoding of hand movements, their interaction with sensory systems. Today, he's going to be speaking to you about work looking at sensory motor networks encoding hand grasping as viewed in the primate brain. The work packages with which Hans's team is involved in for the B-CRATOS project will involve testing a BCI system for control of grasp in primates. So this is today Hans will be going over some foundational work that relates to this task. So Hans, please take it away.
All right, thank you, Paul, for this nice introduction. And thank you to the audience for showing up and being interested in this topic. In this webinar, I will talk about the neurophysiology and the decoding of grasping. That's something that humans and primates are equally well versatile. So if you are looking at how many different grasping movements you do every day, without thinking about it, it's quite immense, right? So you can do brushing your teeth, you're holding pencils and pens in your hand, working with hammers, you're using fingers to point at things, grasping objects, and maybe you play piano or other instruments that are very difficult to operate. And this is also quite an achievement in terms of control, how the brain is actually controlling this. And in particular, since our hand has not always the same size from birth on. So here is, for example, the picture where you see an adult human hand with a baby hand or a kid's hand. And that already tells you that what we have learned in childhood in terms of how to interact with the world has to change throughout your life, because your plant, because your instrument, the hand, is changing its size, but also its configuration and the versatility, the speed, and the materials that actually are working with the operation. So that's quite an achievement for the brain, how to control these movements. And as a motor physiologist, I would be, even more stronger in the point that we have a brain because we move. If you would be a plant or couch potato, you don't need a brain because you're just sitting on the same spot and you just see what happens. And basically you need only, the metabolism taking what's coming in and producing your products. But if you're moving in the world, then you have to interact with the environment. You have to make decisions, what to do, what actions to take at what time. And that's why we have a brain, I would say. And it's very a complex system, and neuroscience is looking into how this works. And what our group is doing is particularly looking at how the hand is working, how the hand is operated by the brain. So that includes taking sensory information in, factoring out or selecting out information that's relevant and maybe ignoring information that's not relevant, and then making choices and decisions on how to interact with the world, and then finally setting out this information to movement activation, so to bring movement into action. And if you look at that in the motor system of grasping movements, we have to look, of course, at the parietal cortex, where sensory information is coming in particular visual information, but also some other sensory information and other senses come in to be integrated. And there is a representation of space and of objects and shapes of objects in the parietal cortex. These areas, and there's area AIP, which is particularly important for grasping, is selective for shape and orientation, as we will see. This area communicates with premotor cortex, which is relevant for preparation of movements, but also interacting with prefrontal cortex on volition. So what is your intention? What is your need? What is the interesting thing you want to do? There also decision-making is forming. And in the end, there is a motor plan generated to select certain grip types to make certain actions. And that information is then processed to motor cortex, primary motor cortex, or M1, where motor selectivities, where motor programs are stored that are then processed and sent down to the spinal cord. Of course, there's modulation by the basal ganglia system, by the cerebellum and the brain stem, thalamus, and so on, which I ignore here for the simplicity. But also, when it comes to motor execution, the sensory feedback, how touch information, how force information of how the hand moves needs to be integrated into the execution of the movement. And this information comes in at the cortical level at primary sensory cortex. And that is important for the guidance of the movement ongoing and for correcting movements. So that also has a big role. And that's also why in the B-CRATOS project, we basically interested in both way communication, how the brain is organizing or is operating movements, and also how it takes sensory information into account. In the context of this presentation, I would like to basically talk about three things. Looking at the grasping system and in primates, and give an overview on how coding is happening in this system. So basically how brain neurons and populations of neurons are active in these different brain areas to generate movements. So that's basically basic systems neuroscience topic to understand how the brain works. And in the second part, we'll then talk about decoding. Decoding means is to some extent the opposite. Basically, when we are reading out information from the brain, from many neurons at the same time, for example, we can ask the question, can we then predict what the brain is doing to control an artificial or later on maybe natural body part to generate this movement? So think about a paralyzed patient who cannot move his limb, maybe locked in patient. This patient can still think about the movement, but is unable to make movements happen. And so if we can interpret brain activity ongoing in real time, while the subject is thinking about doing these movements, we may be able to bypass a blockade or so. And in the third part, I would like to go a little bit more into neural networks. It's a very modern view of looking at population activity, also in different parts of the brain. And the grasping system is a very good example to look at that. We have different cortical areas and different populations in each of these areas, and to ask how do the neural populations functionally connect to each other to make this happen, and what can we say at the moment about it. This is of course ongoing work. In the primate brain, to study the primate brain on grasping, it's very difficult to do this in lower animals like mice or rats, because their movements are quite primitive. They can open and close the paws, but fine-tuned movements like you see here on the power grip or precision grip is very difficult. So that's why our study model is the primate, the macaque monkey. And this can then have a good smooth transition to using recordings in humans that has lately in the last 10, 20 years been done, built on the knowledge that has been gained from working with macaques, with non-human primates. So here you see a monkey a macaque monkey in an experimental setup. He has a little data glove on his finger. I will talk more about that in a moment. And he's interacting with this handle that he's touching with a precision grip or with a power grip in two ways. And then there are some instructions. There's also fixation light here on the top and some extra LED lights that guide the monkey to instruct him which grip type to make. So with this paradigm, and this can go in a little bit more detail, also temporal structure of the paradigm of the task, we can then ask questions on the selectivity of neurons in the brain when we record from neurons in the brain on those properties regarding grasping. And so how that is done, we have like a task where there is rest at the beginning and an instruction what to do, and then the planning phase where the monkey knows what to do but is not allowed to make the movement yet. And then he makes this reach to grasp movement at the end. And then if we record from a neuron, which classically is an electrode inserted in the brain tissue next to a neuron, we can record the extracellular potential, which is actually the voltage over time, which you see on the right side of the display. And then you see low frequency signals, which also includes the local field potential, which I'm not talking about. And then these high frequency spikes overlaid here. And you can make sorting of those spikes. You can look at in more detail to make sure it's only coming from one neuron. But in the end, these are the signs of the action potentials of one particular neuron. And that can be isolated by putting thresholds in and filtering. And then you can identify the times when this particular neuron is being active, meaning producing action potentials. And you see the blue dots here are the action potentials of one neuron over time. And if you then make an experiment, like the monkey is instructed to make a precision grip or a power grip movement. Power grip is like grasping a hammer or a branch, and precision grip is like using two fingers to press on a small object. You can see this one neuron recorded from this parietal area, AIP, anterior intraparietal area, that has is silent in the beginning. So these are spike rasters, the blue dots on the top. Each row is one repetition, and then there are 10, 15 repetitions of the same trial type. And you see the activity, which is then summarized in this curve underneath, is the firing rate over time. This activity is becoming much stronger during the Q phase, when there actually is a light shown, the object is shown. And then this activity goes up and stays up during the planning phase when the light goes off again. So the monkey's in the dark, but he is planning to make the movement. And then when the go cue comes, which is a dimming of the fixation light, then there is the movement executed, and then this neuron is firing an extra second peak at the end of this, and then returns to baseline. So this neuron in AIP is strongly responsive to a precision grip movement. And it has a strong peak during the visual presentation phase, and then the maintaining plateau during the planning phase, and then a second peak during movement execution. When the same neuron is observed during the power grip, so when the monkey makes this branch grasp movement, then the same neuron is only briefly active during the presentation of the object, which is the same for the precision grip in this case, but then goes back to baseline and being silent. So this neuron would be strongly selective or tuned to one particular grip type, right? And then you can try different grip types if you like, as we'll see later. But also you can try different other things. For example, the orientation of this handle can be varied, which we did here. And so another neuron where we see a strong response for precision grip as well, and a weaker response for power grip. But interestingly here, when we do different orientations of the handle, the strongest response comes from the vertical orientation. And then when you turn the handle to the left or turn the handle to the right, then the response is much weaker. So this means this neuron is not only selective for the grip type, but also for the object orientation. So it has a strong selectivity for the object orientation. And you can go on, right? So we can also change the position of the target from being in the center to moving to the left, to the right, or up and down, which is on the left panel here. So the T is the target location in the in red in the middle, and then is shifted to the left and to the right and up and down. And the eye position is the little e that stays at the center. And you see here that the same neuron for the same grip type is profoundly differently active depending on the position of the object in space. That doesn't matter so much during movement execution, but during the planning phase and the cue presentation phase. So that means there is also a selectivity for the location of the object in space, which of course could mean a relationship to the reach, to reach location and to the reach movement, and not for the grip type. But in this grasp area, we see also this modulation of activity depending on the location of the target. And then there is also a grip force variation when you change how much force is needed to grasp. So with a strong grip or a weaker grip or a very weak grip, we see here a modulation where in this case, the neuron is more strongly active for a high force than for a weak force. But some other neurons show the opposite. So there's also selectivity for the grip force. And on the right side, the study shows that neurons in this AIP area, but also in premotor cortex, are also selective for the laterality of the hand. So you show the object and the monkey is instructed to make a power grip or precision grip. And then he should do that either with the left arm or with the right arm. And selectivity for that is quite strong in premotor cortex, but not so much in parietal cortex. So the contralateral activity is strongest present in the premotor cortex. So there's also a selectivity for hand, one way or the other, often contralateral, but not only. And that means we have very many different signals that are present in the selectivity and the activity of individual neurons. So I mentioned, of course, grasping, grasp type, the orientation of the hand, the location in space, grip force, hand laterality. And we could continue on. There is, of course, it depends on how much reward monkey gets for executing the action. And other things like how awake the monkey is, how attentive the monkey is, and so on. So this mixed selectivity shows that the processing of this information in the brain and these brain areas is very complex. And also puts a burden on decoding, because one neuron is not only selecting or is not only representing an individual signal, but a mixture of many signals. And how can you then make use, how can the brain make use of it? In the way that the combination of how much each of these different signals is contributing to the activity of one neuron is varying from neuron to neuron, right? So that means in the average, depending on how you average from a population of neurons, one can very well read out individual representations of target location, grip force, grip type, and so on and so on. But if you just look at one neuron, then you will be lost, because you don't know what exactly is causing this change of activity over time. So if we then look at better understand how the brain processes this information, how the brain is generating these movements, this is looking at grasping movements, but this is pretty much very similar to all kinds of movement. We have to look at this multidimensional process. And that means we need to look at many neurons at the same time. And we need to have a huge variety of objects, not just one object with two grip types, but many different configurations of the hand and many objects. And we need to record for many neurons. So that's what we did in our lab for grasping. And Stefan Schaffelhofer, who was a PhD student in my lab a while ago, he designed an experiment where he built a turntable task. Monkey is sitting in front of this table with a turntable on it and performs a delayed grasping task. And on this carousel, actually multiple carousels, about 50 different objects were presented that were different size and shape and orientation. And he also developed a data glove that can measure the hand shape, the configuration of the fingers and the arm while the monkey is making these movements. So we could then simultaneously measure the kinematics and then record from neurons in the brain, for many neurons in the brain at the same time. The task paradigm is pretty much the same. Basically, there is a fixation epoch, then a cue epoch where the object is shown in the light, and then the light goes off again. And in the subsequent planning phase, about a second or so, then the monkey can plan the movement, but he's only allowed to make the movement when a go signal comes that gives him the go to make the movement. So this way, of course, we can also then look at the different phases of the task where the object is presented in the light, planning is made, and when the movement is executed. This is a short video showing you how this looks and sounds in the monkey setup. So you see monkey is grasping objects in various size and orientation. And the motor in the background turning the turntable. And then this is done randomly, so you cannot predict which object is coming next. At least not which one of the 6 objects on the turntable that is currently mounted is coming up. So with that, we have then about 50 different objects of different size, like, you know, here are cylinders, the cubes, the balls, horizontal cylinders, then, sorry, this is a ring, and then a bar, a vertical cylinder, then objects that look very different, but are of course grasped in a similar way, like with a power grip from above and on the right side, and with a precision grip with the 2 fingers on the left side. And also sometimes the same object or same type of object can be grasped in 2 different ways. So either from above, and one monkey also chose to use a grip from below sometimes, and actually even varied that. So the same object could be grasped in different ways, and the handle on the right also was introduced earlier on, is then also instructed to make grasping movements in different conditions. So to measure then kinematics, we developed this data glove, which is a magnetic-based tracking system with sensors on the fingernails, essentially, and that measures then the joint angles with some calculation, the joint angles of all the finger joints. And if you look at your own hand, you have like 5 fingers normally, and 3 joints for each finger. The base joint can also be moved in 2 degrees of freedom. So that gives you already 4 degrees of freedom per finger, times 5 is 20. And then the hand in space can be translated in 3 directions, shifted in 3 directions, and also rotated in 3 different directions. So that's 6 degrees of freedom of how to position the hand and configure the hand. And then the only degree of freedom that's left is the elevation of the elbow, if you assume the shoulder is fixed in space. So with that, we have then 27 degrees of freedom, which we can track with this hand tracker. And you see here a reconstruction of the hand in this grasping task, where we can then figure out the temporal evolution of the thumb joint angles, the index finger joint angles, the wrist orientation, elbow angle, and shoulder angles as well. So this gives us the possibility to reconstruct the entire movement, as you see on the left, for the different movements, and even apply a musculoskeletal model where you can simulate the length of the different muscles of the hand and arm to make this movement happen. More recently, a small interjection, we also built a new system based on markerless tracking that goes on very modern ways, using a number of about 10 different cameras from different perspectives, and then annotation of the fingers, of the videos of these fingers, to identify the joint angles, to identify the parts of the fingers. And that allows then to reconstruct hand movements even without markers on the finger. And this is available also on our GitHub account, if you like. It's very versatile. It's also very easy to use. Quite a number of labs is using it for their own purposes. And that's kind of going in the direction of Deep Lab Cut, which is a 2-dimensional tool, but this already does 3-dimensional position integration, has a nice tool to integrate different camera positions and for the annotations, and then the computation of the network. So that works quite well. And this is the novel thing we are using in the future. But on the data I presented you, we then used the data glove to make these hand movements trajectories. And if you then take, for example, the static position of holding the object in the hand, you can then classify the object shape, the hand shape, sorry, the shape of the hand configuration for these many different objects. And using a hierarchical clustering analysis, we could actually identify about 20 different classes of grip types for these 50 different objects. And so we can then say these 50 objects of different shape and orientation are grasped with about 20 different grip types. And then we can look at the neural activity for these movements. And to do that, we implanted electrode arrays of metal electrodes. You can see here one enlarged. This is 32 electrodes of various lengths that are implanted in the 3 areas we are interested in. This is the parietal area AIP, premotor area F5, and motor cortex M1, the hand area of motor cortex. This is an intrasurgical picture where you see the insertion process using an electrode inserter tool, and then a cable that connects this electrodes to an outside connector that is mounted on the head post of the animal. And then everything is closed up, the dura is closed, the bone is put back, and everything heals up. And after the recovery of the monkey, after a couple of weeks, you can then start recording and record from about 200 electrodes simultaneously. And you see here an example from AIP where we recorded about 95 neurons with 600 trials. And you can then look at the population activity. Now, this is not many repetitions of the same neuron, but this is one trial of many neurons, right? This is 595 neurons of one trial, and then putting all trials together, you can then group in the neuronal space the population activity. And you don't see, of course, 95 dimensions here, which would be the different axes of a high-dimensional neuronal space to the firing rate of all these neurons. But we see here only from a PCA analysis, only the first 2 and later 3 dimensions of this neuronal space. But already here you see a grouping of the population activity in different clusters. And the clusters are according to the objects in AIP. So you see here grouping with rings and balls and cubes and so on, and not, as you see on the left, according to the hand configuration. So this is going with the object shape. And if we then allow the 3rd dimensions to pop in, you see that this clustering is very, very distinct for these 3 dimensions already. So basically what we see here is that the coding in parietal cortex is primarily on the object shape, object information. And there is more information in the population activity in there, as you will see, because we can also decode like movements from there. But this is in the first 3 dimensions and is predominant. And later on in higher dimensions, you might also see other signals, as I showed you already in a detailed analysis. But the predominant activity in AIP in parietal cortex is visual activity and object information. If we look at premotor activity, and the same would be true for motor cortex, which I don't show here, we can then do the same analysis when we compare now the joint angle space, where we take this 27 degrees of freedom, and from every trial we take the hand shape, and we have then a 27-dimensional vector, and go into the joint angle space and look at the clustering of the grip types, and compare that to the clustering in the neuronal space, where we take the activity of these electrode arrays of these many channels and see again at the neuronal clustering there. And what you see in premotor cortex and motor cortex is actually a very nice similarity between the joint angle space and the neuronal space. And this can be made very systematic and quantitative using a procrustus analysis, which we have done in the corresponding paper. You can also look at the dendrograms, where you look at how similar the different objects are grouping in the joint angle space. So there you will see basically objects that are grasped in the same way are grouping together, like this one and this one. And then the precision grips are here, and the ring is grasped with a vertical power grip, similar to the vertical cylinders. And in the neuronal space, you see basically a similar grouping. And that can be quantified even further in the paper. So to sum up, the neurocortical coding properties in these three areas is such that we find with these 50 objects a coding of the neuronal population according to the shape of these objects and the size of these objects in parietal cortex. And in contrast, in premotor and motor cortex, we find a coding that follows the grip type, so the movement preparation and movement execution. In premotor cortex, we have preparatory activity. In motor cortex, we have predominantly activity kicking in when the movement actually happens. Okay, now the question is, how can we make use of this information for decoding? And basically, we have then the same arrangement that we have neurons in there implanted in cortical areas, and we read out the information, and then we need to send that to a decoder that it makes use of this information as it comes out, so basically in real time. You can do some simulation by doing offline analysis to see how much information is there and how well this can be done. But when it comes to the application of making this work in a patient, then we need to do this in real time, right? Basically, from moment to moment, we have to read out the information and then send out a control signal, for example, to a robotic hand, or as an example, to a cursor for reaching. But in grasping, you would like ideally at least operate a robotic hand, or even better, do some muscles stimulation to get an existing hand back to work. So this has been done not in our lab to start with this is from a video from Andy Schwartz's lab in Pittsburgh, formerly in Arizona and in San Diego a very long time ago. So here he implanted these arrays in motor cortex, and he was able to use a simple population vector decoding algorithm to train a decoder to operate a robotic arm to go to various locations in space and grasp marshmallows. And the monkey was able to control that robot and bring those marshmallows to his mouth and eat it. Right? The main thing here is the operation of the robotic arm. The grip is only very primitive open and closure of a claw there, but that demonstrates the feasibility here. And there has been more work done in patients later on. Here is a video with a human patient that operates now a more versatile robotic hand with a more degrees of freedom in the fingers. And you see here, this person can handle various objects. The grip is still very similar, a power grip, even for the small objects. But the operation of grasping something and moving it in space is working quite well already, right? This looks very nice. But the problem still is the control of the hand, how that is operating. And a final picture from tactile feedback. This is a patient, you see here, he is blindfolded, so he can't see the robot hand and what the experimenter is doing with the robot hand, namely touching it at various locations, various fingers. You see here the implanted connectors on the head where the information comes out. So these are the neuronal signals that are read out through this connector on the skull, and then fed to a computer that interprets these signals in real time. And what this patient is doing in this experiment is just telling which finger was touched on this robot hand, because the experiment is that there's stimulation of
Index.
Primary sensory cortex.
Ring. Pinky.
You see that this works. So
Index.
there is this sensory feedback possible
Middle.
by electrical stimulation of primary sensory cortex
Middle.
to bring back touch information to the
Middle.
brain. This is relevant because if you have been
Ring.
with a dentist, right, then you have a very numb feeling.
Index.
If you got an anesthesia in the tooth or the tongue, you have a very strange feeling in the mouth, but you can still move the mouth and the tongue, but it's a strange feeling, and you cannot really use the tongue and the mouth in a useful way. The same is true if you would inactivate the fingers by numbing the fingers. So tactile sensory feedback is very important. Okay, now what can we do with our setting? We have then these arrays implanted in motor cortex and premotor cortex, and we can then have an analysis of how the population would respond, for example, for precision grip and for power grip. So this is now 4 electrodes by 8 electrode array with different population activity over time. And now we can make decoding by, for example, evaluating the holding phase or the planning epoch of the task and ask how well can we predict the intended movement or the later executed movement of the monkey. And here is a confusion matrix that shows that, where you see the real grip on the x-axis and the decoded grip on the y-axis, and then the correct predictions are on a diagonal. And you see this is about true for 86% of all cases, and errors mainly happen, well, very few actually have doing errors. And you can also look at the contribution of various brain areas. So for the planning phase, for example, motor cortex plays a minor role. Important is premotor cortex, area F5, and parietal cortex to some extent. So this demonstrates that it's possible to predict these 20 different grip types from the recorded population activity in the various cortical areas. You can also go for continuous decoding, and that's what Katharina Menz did, using a Kalman filter to kind of use the continuous tracing of the spiking activity in these populations, and try to reconstruct the kinematics of the arm and hand. So you see in gray the decoded the true kinematics that was recorded with the data glove, and in yellow the decoded kinematics as predicted using the Kalman filter from the neuronal population activity. And you see here there's some wiggling because there's this bigger errors in terms of angle on the shoulder and elbow joint, but the hand shape is doing quite well. So this is a continuous decoding based on the recorded activity in this 50 object grasping task. Of course, this is offline. That means we are just checking that information to make this decode work is there. To make this really work in real time, you need to tell the monkey to think about the movement without doing the movement, because we don't have paralyzed monkeys. And this is work that Andres Agudelo Toro has been doing in our lab, where he built a real-time decoding system. So you see here the monkey and the monkey's hand. And in the initial phase, he's actually making these movements to the handle here, and he's performing a power grip and precision grip. And this is played into a virtual environment. You see this here, where there's the virtual object and the virtual hand. And using the data glove, we can then translate the movement, the kinematic movement of the real hand into the virtual environment, the virtual space. This is just to start. And then the monkey only sees the virtual environment, but not the real hand anymore. And he's just doing this movement in the virtual environment. And then we move on to not use the hand kinematic information to make the movement, as on this slide now, but the neural activity. And then basically you see here that the monkey can do power grip and precision grip based on the neural activity, also the continuous tracing over time. And this needs to be refined and generalized, of course, to include many more grip types and various orientations, various locations in space, and so on and so on. And then we would move on from that to a real robot hand. Using a virtual environment is quite easier to start with, but then when it comes to a real perception of the movement, also including then sensory feedback from tactile information, a real robot hand has a lot of advantages. That's what we are doing that in the context of the B-CRATOS project. We're planning to do that with a real robot hand. The challenges that we have in neuroprosthetics is still the longevity of the implanted electrodes, because there is scarring, granulation tissue growing around the electrodes over time. This is a general thing that happens to all implanted devices or objects in the body. And that is only a problem if you want to record in the neighborhood of a small small neuron. That may cause a problem. That's the main reason I think this technique hasn't been applied in patients a lot. The problem is sensory feedback I mentioned. So that causes basically the need to reverse the decoding, to make stimulation of brain activity, to bring brain activity, to activate the brain in a meaningful way. So we have to understand where exactly in the brain we need to stimulate and how, and then this has to be interpreted in the right way. So that's a new dimension of problems to be worked on. The training of real-time decoders is another issue, because even if you are a paralyzed patient who has been using an arm for a long time, when you're paralyzed, you can't use that anymore, and you don't know the coding principles of these brain areas. So using imagined movements or thinking about movements can help, but then to make a decoder really work effectively over a long time is a challenge that needs to be overcome, and that predominantly will have to be set up and developed. That's why real-time experiments are very important, both in monkeys and later on in humans as well. The idea there is to start with a simple movement, like a reaching out in one dimensions, or a simple power grip or simple grip, where you only control the aperture, and then move on from that to allow more degrees of freedom, like different orientation of the hand, different hand shapes, and so on. That's the idea, and to then have already positive experiences by using this device and then expanding the versatility of the system stepwise. All right, in the final part, which I want to briefly touch, I will talk about neural networks. And using the data set that I described with the three cortical areas, AIP, F5, and M1, Jonathan Michaels used this data to model a recurrent neural network for grasping. So the question there is, can we actually artificially simulate the neural processing that happens in this grasping system, starting from the visual information, so basically giving the system images of the 50 objects, and we want to get out the dynamics of the hand movement grasping that object. So basically the reconstruction of the musculoskeletal model with the changes of the muscle length over time. And what was amazing using the data from the recordings was that using this compartmentalized model with three modules, an input model, an intermediate model, and output model, and various complicated regularization procedures I don't want to go into, it was possible to make this computation from showing visual object features and getting out the muscle velocity signals for the dynamics of the movement. And the actual error about the movement in the end was about 5% for the different joint angles. So that was quite great. But what was even better was that looking at these many artificial neurons and looking at the neurons recorded in the real monkey brain, that they actually matched. And that was not trained in, right? To make this behavior work is something you can train the network to be optimal, to make this error as small as possible. But without telling the system how to make this, we can still see that the actual neural image the neural firing rates and the model firing rates actually looked very similar from a case-by-case basis. And you can do then a better job in doing a canonical correlation analysis to really see what are the features in the population space in the neural network model and the hand model. And that looked like this gives us a better way of understanding how the neural processing goes on in the monkey brain. And that can be now optimized for different subtasks and refined in various ways. Finally, looking at connectivity between the recorded neurons. Of course, it's clear there are thousands and millions of neurons in these cortical areas, and they're somehow connected in a very difficult network. And we record in the order of 200 neurons. So it's very rare that one neuron is directly connected to another neuron with an axon. But what we see is functional connectivity that can go in different steps, like 2-3 steps or so. And you're looking at the cross correlation between the firing rate of one neuron and the firing rate of another neuron. We can see the similarity or the differences, like how often is one spike in one neuron generating or followed by or led by a spike in another neuron. And that analysis was done by Benjamin Dunn using this cross correlation analysis tool. And it's very important to look at the right statistics, make sure the signals are very clean, that there's no noise in there. And then, it's very easy to grade correlation signal, but whether this is really significant or not is very difficult. And he paid a lot of attention on that. And with that, he found basically there are different modules in the 3 cortical areas that have different functions. And so they're connected to each other more closely. And he also found that these correlations, the cross correlations, are sometimes oscillatory, like you see here in the beta band frequency, low frequency correlations like here, or not at all oscillatory. And looking at that, he found basically that there are some neurons are more strongly connected to other neurons, and they form like this extra network of so-called hub neurons, and that is called a rich club. That some small subpopulation of neurons is extremely well connected to these other within this group, within this small group. And then the non-hub neurons, they would have local connections, and then they have also connections to this hub. So if you would correspond, or if you would need to communicate from one area to another, from one local neuron to another, you would need to go first to a hub neuron and then process the information to another hub neuron and then go back to the local neuron. So similar, like when you do a trip to Australia, you first have to go to Frankfurt, take a plane, go to Sydney, and then go into the desert to be where you want to be. So this hub neurons and this rich club makes a lot of connectivity, and the local neurons are much less connected to each other neurons. These hub neurons are strongly oscillatory in different cortical areas with different frequency bands, and the local neurons are very often not oscillatory. And that was a major result that might also explain a lot of the EEG signals that can be recorded from outside of the skull, so that this is a physiological underpinning of the connectivity at the spiking activity at the individual single neuron level across cortical areas, and how that is translating into non-invasive electrophysiological signals in the EEG, for example. This analysis was done mainly taking all spikes of a trial and of a recording together to look at this connectivity. But there might be dynamics, right? There might be in some conditions for some grip types, one connection important, and for another one, another connection important. And this changes in connectivity over time in different phases of the task, like in the cue phase, in the planning phase, and movement phase, and for different conditions. That is something that Swati Sesatri was looking at, and that cannot be easily done with spike-spike analysis, because you need a huge number of trials to do that. And so that's why she used spike LFP analysis using pairwise phase consistency. And with that analysis, she could show that there is temporal dynamics. So that means there are different connectivity patterns present during the fixation, cue, memory, and movement phase. And there is also variation with task conditions. So there's a power grip here and instruction precision grip. You see different patterns of activity. What you see here is a connectivity matrix where you see neurons on the x-axis and the LFP sites on the y-axis, and then a red dot would indicate a certain strength in this pairwise phase consistency number, but normally goes between 0 and 1. You see a strong connection here, AIP, AIP F5 in the beta band, which is missing here on the slide, but this is the beta band activity in this frequency band. And that is slightly different for power grip and precision grip. So that gives us way to better understand the connectivity at a functional level. I think I stop here and summarize. What I was talking about was that cortical areas contribute differently to the control of hand movements, with parietal cortex, in particular area AIP, anterior intra-parietal area, that presents predominantly object information. Premotor cortex, and particular F5 for grasping, encodes grip type intentions. So that's plans to make grip type movements. And motor cortex represents best for the accurately movement execution. So it becomes strongly active when the movement actually happens. And we find in all these areas mixed coding, especially in AIP and F5, of a multitude of signals in single cortical areas. Looking at population activity from the grasping network, we can see and demonstrate that it can be used for decoding grip types and hand kinematics. So both categorical and as well as parametric decoding. It can be explained by a recurrent neural network model, which of course then can be refined. And network connectivity analysis of the grasping network demonstrated a rich club topology of highly connected hub neurons. So that's a backbone, and oscillatory hub connections in the beta and low frequency range, in contrast to, for example, gamma activity if we look at visual areas. So that's also the frequencies seem to be specific for at least for the cortical areas. Pairwise phase consistency analysis reveals condition and temporal variation of this functional connectivity, which of course is to be expected on the dynamics of the network. So thank you very much for your attention. Here is also the people in the lab that contributed to this work. Thanks for your attention. I'm open to questions.
Thank you, Hans. So that was a really nice overview of kind of, I think, a lot of the foundation for what we're going to try to do with B-CRATOS in terms of decoding for grasp and building a BCI demonstrator. Does anybody, I noticed there were no questions added to the chat during the talk, but if anybody has a question, please raise your hand or jump in. We have about 5 minutes. So does anybody have any questions for Hans? So Hans, I have a question for you. I think in the sense of what we're trying to accomplish with B-CRATOS in terms of what we're looking to demonstrate, we're trying to move large amounts of data that would allow you to extract spiking information from many channels to decode for some grasping movements, some simple control of a BCI of a prosthetic. In the ideal case, how many channels do you truly need with sort of the right kind of signals to control a couple degrees of freedom? Is it really, do you feel like a BCI designed to allow someone to say maybe do something like cursor control and clicking, channel count is the most important thing, or is it more about the quality of the data? in the short term, but also over the long term? What ideally are you getting from the device in terms of signals?
Okay.
It's a pretty big question, but I just
yeah, but it's an
wondering.
important one, right? So let's see whether I can show a relevant slide here. From maybe this one. No, this one. Sorry. So from the I guess you can do with a very small number of neurons if the recording quality of that neuron, those neurons, is very high, and that might be variable. So in the beginning, if you're in the right spot and you might get good signals, you might have like 60, 80 neurons from an array of 90 channels or something like that, or even less. That might go down because of the encapsulation of the electrodes. But very often also the recording quality is less than optimal. And then it depends a little bit on the diversity of the recordings and the signal to noise ratio. And then when you want to do only like distinction between power grip and precision grip as a category, you can get away with very small number, right? Like maybe 10 neurons or so could be already quite good. When you want to do more refined movements of also controlling the direction of the hand or the wrist orientation, and more fine control or even temporal changes, like kinematic, parametric changes, that is a different story where you need more neurons. In the end, I guess, if you have a good set of population activity, you could do a nice dimensionality reduction like PCA or something similar than that. You probably are okay with a very few number of components or hidden channels, like 10 or so. But you have to start with a certain larger number of neurons, and something like 100 neurons per area, I think, is a good start to keep going. And from our experience, the signal quality is often very good in motor cortex because the neurons are very big and easy to record. Also, the location of the hand area goes down a little bit on the side, but it's easy accessible. Premotor cortex and parietal cortex can be much more difficult to access for some strange reasons, at least in the monkey. That can also contribute to different success rates in the recording. So yeah, it's a good idea to have a decent number of neurons to start with this enterprise. And then over time, it was reported that neurons come and go, right? You have then changes of the electrodes in relationship to the brain, even when it's embedded, but maybe there's a bump on the cage or something else. And then neurons come and go. It's a dynamic system. And then you might lose a neuron, but you get another one, and that has different properties, selectivities, for example. And that needs to be then adjusted in the population analysis as well. So that's a different kind of problem.
We have another question. Thank you, Hans. We do have another question from Chadwick Boulet. I guess I can read it just for the sake of expediency. Chad says, I know that Jonathan had to do a lot of simulation work to prove that the correlations he was seeing between the artificial neurons and the real neurons was not simply spurious, but in my opinion, the correlations weren't amazingly stronger than what you could see with random activities. Do you think that better neural network models, maybe more biologically inspired, spiking, etc., might allow one to solve the tasks with lower number of parameters, and will those correlate well with real neural signals? And maybe you can read it in the chat as well.
Yes, of course, I was skimming a lot about different things that Jonathan tried in this paper. Getting some output that is accurate, like this 5% thing is easy to achieve because you just have to train a network. You can also do this with a one compartment network. In the end, it's a neural network that you optimize to do the task. So in some sense, we wanted to have it naturalistic in the way that we have a sparse connectivity between the modules using 3 modules. And he was using different assumptions to test out whether this had a strong influence on the neuronal population and how that would compare to the recorded neurons in a monkey. So some of that was definitely intended to be testing out the similarities, and some of it came out by chance, I would say. Regularity parameters like cost on firing rate, weight cost on input-output connections, and sensitivity on complex state trajectories also have to do with the fact that in the recurrent neural network model, which is of course a linear model with some nonlinear transients, they can do easily with negative numbers, but real spiking activity is of course non-negative, right? We have spikes that go back down to 0 Hertz activity and go up to some 100 Hertz or so, maybe 200 sometimes, but not negative. And that is something the network would need to work in a different way, not in the same way as the normal mathematics of networks would work. And that has to do, and then those regulatory things have to do with that, to make the math not too simple, to allow similarities. But it's true, it's an art that traditionally means a lot of tweaking until you see the results you like. And it's also a form of honesty to tell that you're not tweaking and then just show in a circular fashion what you found, because you have doing the tweaking enough. So it's important to say what was tweaked and what was basically then just observed.
Chad, do you have any follow-up? Okay, he says no, but thank you.
Thank you.
Okay, I think we've gone a bit over the hour limit. So if anybody else has any follow-up questions for Hans, his contact information, I believe, was included in one of the slides. Also, just feel free to reach out to us through the website, and we can forward it along. Thank you so much, Hans, for sharing this talk with us.
Well, thank you for having me.
Yep. And I think I will end the webinar now. Thank you so much. Bye, everyone.